Hello, you're welcome to Medical Sciences Medicine by Naphtali Muhumuza. Today we want to look at inflammation. Inflammation is part of pathology and sometimes it is covered under histopathology. So we want to see how inflammation plays a protective role. Some people think it is a disease, but it is a protective role. It is protecting our body from exogenous or endogenous pathogens. And this inflammation comes from a Greek word inflammea. Comes from a Greek word called inflammea. And this inflammea means to burn, or what we call fire burning. So you find that the surface or at the site of inflammation there is burning cessation. That's why the word inflammation came from. And this inflammation is a local vascular response. Is a local response, vascular response to injury. So we see blood vessels responding in the presence of an injury. And this injury can be caused by many factors. So causes of this injury, which can be exogenous or endogenous, include agents like biological agents. We can see physical agents. We can see chemical agents. We can even see immunological changes, and sometimes metabolic disorders. Metabolic disorders also coming into play. Whereby under metabolic disorder, we can see guys like diabetes causing this inflammatory response. Then we can immunological, we see autoimmune diseases doing the role. Autoimmune disease, we see the complement system when activated doing the role of, so antigen antibody reactions. Whenever they happen in the body, we see hypersensitivity reactions triggering inflammation in the body. Then chemical, we see some drugs triggering when we take some drugs, some toxins. Toxins may be snake venoms. You can see bites triggering inflammation. Then physical, we can see increase in temperature, increase in pressure. We can see radiations like X-ray radiations, UV radiations from the sun can also trigger inflammation. And then biological causes, we see bacteria doing that, we see viruses, we see fungi doing that, and parasites. So these are biological. So these are the causes of inflammation, which can be inside the body, what we call endogenous causes or exogenous causes triggering local vascular response to this tissue injury. That is what we call inflammation. And we said this inflammation it plays a protective role. Its significance, it plays a protective role in preventing further injury at the site, and also it plays a big role to kill the pathogen. At the end, we shall see killing of the invading pathogen or the causing agent being killed to prevent further injury to the tissue site. This is what we call, why we say inflammation is not a disease, but is a protective role. And after seeing the causes, the next thing we can talk about is the types of inflammation. Types or classes of inflammation. And this inflammation is divided into two, whereby we have acute and chronic inflammation. Acute inflammation, it occurs faster, and its duration is short. It has a short duration, and the major cells which are found in acute, we shall see the key player being neutrophils. Then in chronic inflammation, you see it takes long duration, that is a slow process, and the cells which participate in chronic, we shall see that lymphocytes and monocytes playing a great role in chronic inflammation. But today we want to focus on acute inflammation. We want to see how does acute inflammation occur and what are the events that occur. And we are going to see that under acute inflammation, we are going to see that it involves two events, whereby it involves the vascular events, and we have vascular event and cellular events. And these events under vascular, we shall see transmigration, that is migration, transmigration, we shall see transmigration. But before we talk about transmigration, let's talk. In here we shall see migration, rolling, and adhesion of the cells. After adhering, we shall see transmigration, and this transmigration occurs by diapedesis. Then after transmigration, we shall see chemotaxis, and lastly we shall see phagocytosis. These are cellular events that occur during acute inflammation, whereas during vascular events, under vascular events, we are going to look at a moment of vasoconstriction followed by vasodilation, and then vasodilation will follow stasis and increased vascular permeability. Increased vascular permeability. So these are the vascular events. So we are going to see how do vascular events under acute inflammation come about, and how does cellular events come about. As we have said, that this acute inflammation is faster, but it takes minutes to hours. It takes minutes to hours, and the cell which does great work is the neutrophils. So we're going to see how vascular events come up and how cellular events come up. But we want to begin with vascular events. And we want to say that if the site of inflammation is here, at the site of inflammation, we see mast cells. These are what we call mast. In the tissue, we find there mast cells. And these mast cells, these are the mast cells in the tissue. And this mast cell, in the case of the trigger of inflammation, this mast cells, it is triggered whereby the IgE comes and binds. And after binding, it causes release of inflammatory mediators. We shall see release of histamine, release of serotonin, release of prostaglandin E2, shall see release of nitric oxide, release of leukotriene B4, release of cytokines like interleukin 8, release of thromboxane A2, and many others. So there are many inflammatory mediators which are released when mast cells are triggered to release these inflammatory mediators. And we want to see what causes a moment of vasoconstriction. That whenever the acute inflammation is triggered, we are going to see production of histamine, production of serotonin, and it is this serotonin that causes vasoconstriction. That the blood vessels narrow their lumen size, they become smaller. And that is what we are calling a moment of vasoconstriction. We are on vascular events. Vascular events, we are saying a moment or in a second we see vasodilation. And this vasodilation is being, the key player is serotonin, being produced by the mast cells at the site of injury. But whenever the site of injury, we see there is trigger of production of serotonin, and thromboxane A2. Serotonin and thromboxane A2, which are strong vasoconstrictors. So they cause a moment. It is a neural change that occurs that whenever there is an injury at the site, the vessels first constrict for seconds, then it is followed by vasodilation. And this vasodilation is triggered by inflammatory mediators like nitric oxide, like histamine, and prostaglandin E2. These are the ones that trigger vasodilation. That after vasoconstricting to become narrow, then it is followed by vasodilation, where the same blood vessel increases the lumen size. So this is vasoconstriction, this is vasodilation. And we have seen the key players under each event. And whenever there is vasodilation, there is increase in the blood flow. So we see increase in the blood flow coming at the injured site, carrying many blood cells. And these cells include neutrophils. And when these neutrophils are brought in here, we shall see that after vasodilation, it will be followed by stasis. Stasis, that is decrease in flow of blood. What decreases the flow of blood is vasodilation. Increase the blood flow to the site, then the blood starts to stop flowing. That is what we call stasis. And whenever stasis comes in, the other inflammatory mediators produced here, like prostaglandin E2 and bradykinins, which will cause what we call increase together with histamine. These ones, they trigger what we call or increase vascular permeability. Vascular permeability. And this vascular permeability is, they start creating pores in the vascular wall. So the endothelial layer of the blood vessels, it creates walls, it becomes permeable, so that we start seeing fluid flowing into the extracellular matrix due to increased vascular permeability made by the bradykinins and the histamines. So that is what we call increase in vascular permeability, which causes leakage of the intracellular content into the extracellular space. And whenever they leak into the extracellular space, this is what leads to what we call swelling. This one leads to swelling, which is one of the cardinal signs. Swelling brought about by increased vascular permeability, leading to the migration of intracellular fluid into the extracellular matrix, causing swelling. Then we see the bradykinin, the prostaglandins, that whenever there is increased of vasodilation, it increases the blood flow to the site, and this increase in the blood flow causes hyperemia, and hyperemia is seen as reddening of the site. So we see redness, the site becoming red because of increased blood flow to the injured site. That is another cardinal sign of the site being red. And because we are seeing also presence of prostaglandin E2, which stimulates the pain center in the brain, leading to development of pain, which we call DORA. Pain is DORA. Then we see swelling being tumor and reddening being rubber. Those are the Greek words. So after these vascular changes, whereby we have seen that whenever there is something triggering acute inflammation, the first thing is vasoconstriction, followed by vasodilation. And when vasodilation occurs, we see increased blood flow to the injured site, causing reddening of the site. And when there is increased blood flow, meaning there is delivery of many blood cells, including the neutrophils. And these neutrophils, we are going to see how they are going to play a big role in completing completing what we call acute inflammation. And we have seen also under vascular events that after the vessels have vasodilated, we see stasis or slow moving of blood. And when the blood slows in movement, we see it will facilitate the cells to start adhering to the endothelial cells. And when it starts to slow, we see these inflammatory mediators like bradykinins and histamines causing walls or holes within the vascula or the endothelium of blood vessels, causing increased vascular permeability. So now from here we can go into what we call cellular changes. These cellular changes, which are occurring during acute inflammation, cellular events, which I've seen, the first one is migration, rolling, and adhesion of the neutrophils. We have what enables migration is that migration is the movement of the neutrophils from the center towards the margin of the endothelium. So when they move from the center to the margin, that is what we're calling migration. And these neutrophils have the selectins and integrins, whereby it has the P-selectin and the endothelium has the E-selectin and the integrins. And the integrins, so we are saying the key players in this process of migration, rolling, and adhesion, we have the selectins and the integrins. Whereby the selectins, we have the P-selectin and the E-selectin, what we call endothelium. Then under integrins, we have the VCAM type 1. Then we have also the ICAM type 1. These are the integrins that we use. Whereby the VCAM, what we call vesicular cell adhesion molecules, type 1, is responsible for the migration, early phases of migration and rolling, what we call pavementing, rolling and pavementing. Then we see the ICAM, which is intracellular adhesion molecule, playing a role in the adhesion of neutrophils. So these are the functions of the integrin. Then the P-selectin found on the surface of neutrophils is the E-selectin found on the endothelium of the blood vessels. So these are the guys that help us in migration, rolling, pavementing, and adhesion of cells. But what happens is that when the neutrophil from the center migrates to the surface of the endothelial cell, we are going to see it is going to use a vesicular membrane known as the P-selectin to bind to the E-selectin. This is the E-selectin. And when this E-selectin binds, it starts rolling. It starts to roll along the membrane, and as it is rolling, it binds with the VCAM. We have also the VCAM, then it is rolling on it, and it continues rolling along the membrane of the endothelium, whereby it is here rolling in the presence of the VCAM, then lastly it attaches itself on the, so this one we are going to call it ICAM type 1, then this one was the VCAM type 1, which are enabling rolling. We said the VCAM enables rolling and pavementing, then ICAM, for it enables adhesion. So we are going to see that these neutrophils, they now by the time they reach here, they have adhered. They adhere firmly. What enables them to adhere firmly to the endothelial surface is what we call the ICAM, what we call intracellular adhesion molecules. These intracellular adhesion molecules enable firm attachment of the neutrophils together with the endothelial surface. And after touching firmly, we shall see it undergoing what we call transmigration. They will undergo transmigration. Whereby this neutrophil will force itself by diapedesis and forces itself to pass through the vesicular pores, created by the histamines and the bradykinins. And this flow of neutrophil is what we call transmigration and occurs by diapedesis, whereby it squeezes itself in the small pores created in the vesicular walls, leading to transmigration. So this is what we know as transmigration of the neutrophils from inside the cell into the extracellular. So we shall find the neutrophil in the extracellular matrix after transmigration. So this is transmigration by diapedesis. And after reaching in the intracellular space or extracellular matrix, or you can call it in the interstitial space, we see this is the site of inflammation. And this site of inflammation is producing substances like leukotriene B4 and interleukin 8. So we are going to see leukotriene B4, we are going to see interleukin 8 causing unidirectional flow of the neutrophil towards the inflammatory site. So the neutrophils start moving towards the inflammatory site in one direction, enabled by the chemotactic factors. These are what we enables chemotaxis. So after transmigration, there is unidirectional flow to the enemy. And what tells the neutrophils that the enemy is this side is these mediators like leukotriene B4 and interleukin 8, which play a big chemotactic response, together with complement 5A and complement 3A. We know that complement 5A and complement 3A also they are chemotactic. So leukotriene B4, interleukin 8, complement 5A are chemotactic factors that enable the flow of, or the movement, unidirectional movement of neutrophils from the vesicular extracellular space towards the inflammatory site. And what has enabled them to move towards by unidirectional flow are leukotriene, interleukin 8, and complement 5, which are chemotactic responses or chemotactic substances. So we have seen migration, rolling, and adhesion. Then the second one was transmigration. And we have seen this transmigration is by diapedesis. Then the third one, we have seen chemotactic flow, chemotaxis. And this chemotaxis flow of the neutrophil towards the injured site has been guided by leukotriene B4, been guided by interleukin 8, by complement 5A, being chemotactic factors. And after this, the fourth one is phagocytosis. And this phagocytosis, which is the last part of acute inflammation, it is divided into what we call recognition and adhesion and attachment, recognition and attachment. So the neutrophils, after coming, they have to recognize that this is the injured site and they have to attach. And what enhances this is that we have complement 3B, which acts as an opsonin. So this complement 3B comes on the surface of cells at the injured site and tags or marks that this is where the enemy is. So the one that marks the enemy is complement 3B, which make the neutrophils to recognize the pathogen. And after recognizing that this is the pathogen or the enemy, and that's when we shall see engulfing. We shall see engulfment, and this engulfment is by the neutrophils. After they have recognized, they attach themselves on the pathogens and they engulf. And how do they engulf? Is that if I have a neutrophil, it forms the pseudopods. The neutrophil which was round forms false mouth. it changes its shape and forms what we call pseudopods. And these pseudopods, they are false mouth. So in case the bacteria is in front of it, it engulfs the bacteria at the injured site. After engulfing, it continues to form a phagosome. We find our neutrophil, it forms a phagosome inside. This is the nucleus. Then we find the bacteria inside what we call a phagosome. This is the phagosome, and after reaching to form a phagosome, which contains the bacteria which were triggering the inflammation, we see lysosomes also coming to join. These are the lysosomes. And these lysosomes, when lysosomes are released, they are scissors, they enter and begin killing the bacteria. And this one which has been formed, the combination of a phagosome together with the lysosome, is what we call phagolysosome. So this phagolysosome, these lysosomes enter and starts cutting or breaking down the bacteria, killing them into portions. That is one way of killing the pathogen. And if we don't kill them by forming a phagolysosome, we can also kill them when we form a phagosome inside the cell, it forms what we call reactive species. And these reactive oxygen species, we call them reactive oxygen species, which include hydrogen peroxide, which include hypochlorous acid. That the body or these neutrophils form hydrogen peroxide, which gives rise to superoxides. It gives rise to superoxides, or what we call superoxides. And superoxides can give rise to Gic, which we call hypochlorous, and this Gic kills the pathogen together with the whole cell. That's why we see at the inflammatory place, we find their pus cells. But these neutrophils, after killing the enemy, they produce Gic, which is suicidal. So as they are killing the enemy, they are also killed at the long run, making what we call pus cells. So for them, they kill the pathogen, and themselves, they surrender themselves as a ransom for us to survive. So this is what we can talk about under acute inflammation. At the beginning, we have told you what is inflammation. We have seen the causes of inflammation. We have seen the classification of inflammation. We have seen it is acute, which is the first, and duration is short, and the neutrophils being the key players of the acute inflammation. We saw chronic, which we shall talk about in another video. For it is slow, it takes long, and the monocytes and lymphocytes are the cells that are involved. And we have seen under acute that it has two events, what we call vascular events and cellular events. And we have seen how vessels undergo first a moment of vasoconstriction, followed by vasodilation, stasis, and increased vascular permeability, leading to production of exudate, whereby the intracellular fluid was moving into the extracellular space, causing what we call exudate. So the swelling which is brought about in the extracellular space is due to the accumulation of exudate, which is full of plasma proteins and cells, and it is always red in color. That is what we call exudate, which is different from transudate. And from there we have seen cellular changes, whereby in undercellular changes we have seen migration, rolling, and adhesion enabled by selectins and integrins. And we have seen the roles of all selectins and integrins, whereby the VCAM is doing the work of migration and rolling and pavementing, whereas ICAM, which is intracellular adhesion molecule, leading to firm attachment, which results into transmigration, which is the diapedesis, then followed by chemotaxis, and we have seen chemotaxic factors that enable the directional flow of the neutrophil towards the pathogen. And after reaching there, these phagocytosis starts, whereby it starts with recognition and attachment. And how do we recognize? We have an opsonin known as complement 3B, and after marking the pathogen, the neutrophils come and attach and engulf by forming false mouth, what we have called pseudopods. And then when they engulf, they form a phagosome, whereby a phagosome, lysosomes come and combine to form a phagolysosome, which kills the pathogen. Or this can make reactive oxygen species like hydrogen peroxide, which forms superoxides. And these superoxides can form GIC, and this GIC kills the pathogen together with the neutrophil, leading to formation of pus at the inflammatory site. As we end, we can talk about the cardinal signs, whereby cardinal signs of inflammation, cardinal signs is reddening, we have pain, we have swelling, we have pain, swelling, then we have loss of function, and reddening is brought about by increased blood flow. When there is increased blood flow, we see more oxygenated blood being brought, which is red in color, causing reddening, which we know as rubor. This is rubor. And then pain we saw it is brought about by prostaglandin E2, which stimulates the pain center in the brain and causing pain. And this pain is known as DORA. You can call it DORA. Then swelling, which is known as tumor, is brought about by the exudate after increased cellular permeability, whereby exudate in the extracellular matrix leads to swelling. Then we also can result into loss of function. So these are some of the cardinal signs of, and apart from reddening, pain, we have seen swelling, we have seen loss of function, and many others which we can talk about. Thank you so much for following. This has been acute inflammation. We are always privileged to have you at Medical Sciences Made Easy by Naphtali. Always remember to subscribe, like the video, and share your comments. Thank you so much. In the next video, we shall look at chronic inflammation.